Figure 1 shows a human heart, and let's just do a quick recap of what's happening in the heart. So we've got the oxygenated blood from the lungs entering the left atrium, and then the left atrium contracts and squeezes the blood into the left ventricle. Then the left ventricle will contract and push the blood out of the aorta to the body. So the body's got a lovely supply of oxygenated blood. And on the right side of the heart, remember, it's the person you're looking at's left and right, not your own. So on the right side of the heart, we've got deoxygenated blood coming back to the heart from the body into the right atrium. Then the right atrium squeezes, contracts, and pushes the blood into the right ventricle and then out through the pulmonary artery, taking the blood to the lungs to oxygenate up. There's an easy way to remember which blood vessels take blood away from the heart and which blood vessels take blood to the heart, and that is that arteries take blood away, and they both begin with A, and veins take blood into the heart, and in veins you've got ins, so it reminds you into the heart. Which blood vessel carries deoxygenated blood away from the heart and to the lungs? Is it A, B, C or D? So we don't actually even have to remember the names of them, and we've already drawn it out, so we know it's B. The natural resting heart rate is controlled by a group of cells that act as a pacemaker. Where in the heart are the pacemaker cells found? Well, they are found in the right atrium. Some people may be treated with a drug to slow their heart rate. Digitalis is a drug that slows the heart rate. Where does the drug Digitalis originate from? Well, this is actually from those pretty flowers here that are called foxgloves. Beta blockers are another type of drug that slows the heart rate. Table 1 shows information for people who do not take beta blockers and for people who do take beta blockers. Stroke volume is the volume of blood pumped out of the heart each time it beats. Cardiac output is the total volume of blood pumped out of the heart each minute. Calculate the stroke volume X. Okay, so that's our missing bit in the table, and they've given us an equation. How nice is that? And give your answer to two significant figures. So the cardiac output is 2,800. We don't know what the stroke volume is. We've got to work that out. And we know the heart rate is 52. So what we've got to do to work out our stroke volume, which is the volume of blood pumped out of the heart each time it beats, we've got to divide the cardiac output by the number of beats. So 2,800 divided by 52, that gives us 53.846153. Don't forget, we've got to put that in two significant figures, so that is going to be 54 centimetres cubed. Some people who take beta blockers get out of breath when they exercise. Explain why beta blockers can have this effect during exercise. You should refer to the information given in Table 1. So if we have a look at the table, when no beta blockers are taken, 18,000 centimetres cubed of blood is pumped from the heart every minute. But when someone is exercising while taking beta blockers, the volume of blood is significantly less. So that means there's going to be less oxygen and glucose delivered to the working muscles. So less aerobic respiration is going to happen. It's going to be lactic acid buildup. It's going to be oxygen debt. So this is a 6 marker, so we will need to add as many key points as possible. When a person exercises, they require more energy. Therefore, more respiration is needed to release more energy to the muscles. This means that more oxygen and glucose will be needed. Beta blockers reduce the heart rate during exercise, so less oxygenated blood will reach the muscle cells. Anaerobic respiration is going to occur, and there will be an oxygen debt to those muscle cells. A reduced heart rate and reduced stroke volume leads to a reduced cardiac output, meaning less blood will reach the muscle cells, leading to further oxygen debt and buildup of lactic acid. To get more oxygen into the body, breathing rate and depth will increase, which will also help to remove carbon dioxide from the body as well. Now, to get the 6 marks, there are 3 key areas you need to have mentioned in your answer, which we have, but I've written them down so you remember. You have to have mentioned respiration, the effect of the beta blockers, and the effect on breathing rate. Number 2, this question is about digestion. Name the enzyme that digests starch in the human digestive system, and that is amylase. A student set up a model to represent the digestion and absorption of food molecules in the digestive system. Figure 2 shows the student's model. The method is fill a test tube with water at 37 degrees C. Test the water for starch and for sugar. Mix together starch and enzyme solution and immediately test it for starch and sugar. Fill some partially permeable tubing with the starch and enzyme mixture. Seal the tubing and place it in the test tube of water. Place the test tube in a water bath at 37 degrees C. After 30 minutes, test the mixture inside the partially permeable tubing and test the water in the test tube for starch and sugar. We need to remember that starch is made up of lots of sugar or glucose molecules bonded together. And if the enzyme works that's inside that partially permeable tubing, then that is going to break the starch down into the little individual glucose molecules. And then these little individual glucose molecules will be small enough to pass through the partially permeable membrane and into the water in the test tube. Suggest which parts of the body the partially permeable tubing and the water in the test tube represent. Well, the partially permeable tubing is going to represent the small intestine, and the water in the test tube is the blood. So we got 4 tests, different descriptions of the liquid and results of starch and results of sugar test. Name the reagents used to test starch and for sugar. Starch is tested using iodine solution. Remember, it's that brown solution, and if starch is present, it changes to that blue-black colour. To test for sugar, we have to heat with Benedict's solution in a water bath. Why was there no sugar present in test 1? At the start of the experiment, none of the starch has been broken down yet, or the enzyme hasn't started to work. So we are not going to find any sugar in there at the moment. Explain the results for test 3. The mixture inside the tubing after 30 minutes, positive for starch and a positive result for sugar. So that means that inside that tubing, the enzyme started to work. Some of the starch has been broken down into its glucose and some of it hasn't been broken down. So we're going to get both starch and glucose or sugar present in that tubing. Explain the results for test 4, the water in the test tube after 30 minutes. So we're looking at the test tube, not inside the tubing. We have a negative for starch and we have a positive for sugar. Starch is too big to pass through the partially permeable membrane into the water. But sugar, or glucose, is small enough so it can diffuse from the bag and into the water. Question 3. A student used a pedometer to investigate the rate of water uptake in a plant shoot. This is what a pedometer looks like. So just a quick recap here. As the water evaporates from the leaves in the shoot, that is going to draw more water up from this capillary tube, and we've got a little air bubble in there. So as the water gets taken up into the planty shoot to replace the water that's evaporated, then the air bubble is going to move along. The rate of water uptake is the distance the air bubble moves in a given time. This is the method used to place the pedometer in moist air at 25 degrees C. Position the air bubble at 0 millimeters in the capillary tube. Record the position of the air bubble in the capillary tube every minute for 5 minutes. Repeat steps 2 to 3 with the pedometer in different conditions. Table 3 shows the conditions used. Investigation A, B, C, we've got moist air at 25 degrees C. dry air at 15 degrees C, dry air at 25 degrees C. After investigation A, the air bubble had moved part way along the capillary tube, suggests how the student moved the air bubble back to 0 millimeters for the start of investigation B. Well, all you've got to do is open the tap to allow the water from the reservoir to push the bubble back. Capillary tubing is very narrow. Explain why narrow tubing was used. The air bubble will move further in a given time if you've got a really tiny narrow tube, and it will be easier to see very small changes, and that means the resolution is improved. Figure 4 shows the results for investigation A. The cross-sectional area of the capillary tube was 0.8 millimeters squared. Calculate the rate of water uptake for the investigation A in millimeters cubed per minute. So we've got to use our graph. So if you go across the x-axis, 5 minutes, go up to the line, and you can see that that line is at 66 millimeters. So 66 millimeters is how far the bubbles moved in 5 minutes. We need to make that into per minute, so let's divide that by 5. 13.2 millimeters per minute is the distance the bubble's moving. Then we need to times that by the area of the capillary tube, which is 0.8 millimeters squared, and we get 10.56 millimeters cubed per minute. Table 4 shows the results from investigation B, and I'll just remind myself, investigation B is dry air at 15 degrees C. Plot the data from table 4 onto figure 4. You should draw a line of best fit. Now remember, that line can be a curve of best fit or a straight line, and then label the line B. You need to use a sharp pencil, and you need to do neat crosses. 0 is 0, 1 is 6, and these squares are going up in 2, so that's nice and easy. At 2 minutes, we've got 16, 3, 22, 4, 30, and 5, 42. On my graph, it looks like that 2 minutes, it's slightly sitting off. Now, it is quite difficult for me to do an accurate line of best fit on this tablet that I'm using, but I feel like if I do a slight curve, I'm going to get more points in. So I'm going to do a curve of best fit. There we go. And that is my line B. Investigation C was carried out in dry air at 25 degrees C. Draw a line on figure 4 to show the results you would expect for investigation C. So we need to estimate dry air at 25 degrees C. We know that should be faster than dry air at 15 degrees C, because we increase the temperature, we're going to be increasing the evaporation from the leaves, so that's going to be pulling up water more quickly and moving the bubble along. And if it's dry air, then there's going to be a steeper concentration gradient, and so evaporation is going to happen more quickly into dry air than it would moist air. So we would put our line, just make sure it's above A basically, because it's going to go faster than A and B. The investigations were carried out in daylight. The air bubble would not move if the investigations were done in the dark. Explain why. There's no photosynthesis, therefore no stomata will be open, so there will be very little transpiration, which of course is the evaporation of the water from the leaves. Pathogens are microorganisms that cause infectious diseases. What type of pathogen causes malaria? Well, that is definitely a protist, and if you remember, these are found in the salivary glands of mosquitoes, and when mosquitoes bite a human and insert their little mouthpiece in to get a lovely bit of blood, they also insert the protist into the human, and then that protist starts to carry out its life cycle and cause malaria in the individual. Give 2 methods used to prevent people catching malaria. Give a reason why each method works. So method 1, I think, is the most obvious one. That would be to use mosquito nets at night time. This will avoid being bitten while you're sleeping. You could use an insect repellent, and that means that you're less likely to be bitten. You have only been asked to put 2, but you could also have put insecticides, which will actually kill the mosquitoes. There are also vaccinations now, and you could take anti-malarial tablets as well. Describe 2 differences between a bacterial cell and a eukaryotic cell. A bacterial cell is also a prokaryotic cell. So one difference would be that a bacterial cell doesn't have a nucleus, and another one would be that a bacterial cell has plasmids. You might have also put bacterial cell is smaller or do not have mitochondria. A scientist investigated the population growth of bacteria in a culture solution. At the start of the investigation, the culture solution contained all the nutrients the bacteria needed. The scientists determined the number of living bacterial cells in the solution every hour over 2 days. Figure 5 shows the apparatus used. So we've got a culture bottle, culture solution, and cap with air holes. So let's have a quick think about this before we answer the question. If we've got a cap with air holes in it, then that's going to allow oxygen in for the bacteria and their carbon dioxide out. And the culture solution, that means you've got the bacteria and they're also surrounded by nutrients such as glucose. Describe why there are air holes in the cap of the culture bottle. Well, we've already had a look at this. And it is so the bacteria can respire aerobically. Figure 6 shows the scientists' results. So we've got the number of bacterial cells per centimetres cubed and time in hours. Give one reason for what is happening to the number of bacteria at each of the stages. Well, in stage A, the bacteria are adjusting to the environment. So there's limited cell division, or also known as binary fission. For stage B, we can see that there is a rapid increase in the number of bacterial cells. And that is because the cells are dividing rapidly because there's a good supply of food. And let's give an example, glucose. At stage C, we can see that the rate of cell division is equal to that of the bacterial cell death. So the number of live bacterial cells stays the same. And at stage D, the bacterial cells are dying due to the nutrients and the glucose running out. Figure 7 shows 2 cubes. Cube X represents a bacterial cell and cube Y represents a small multicellular organism. A bacterial cell can absorb all the nutrients it needs by diffusion through its outer surface. Explain why a multicellular organism cannot absorb all the nutrients it needs by diffusion through its outer surface. You must include calculations in your answer. So you can see that this cube X has a bigger surface area compared to its own volume. And you can see that cube Y has a smaller surface area compared to its own volume, and it's certainly smaller, that surface area to volume, than cube X. But we're going to do the maths to prove it. So let's start with the surface area to volume ratio for X. We've been given the measurement 0.002. We know that we need to find the area of one side of the cube. So that's 0.002 times 0.0002, which equals 0.0000004. And then we know we have to times that by 6 to give us the total surface area of the whole cube. So that is going to be 0.0000024. And of course you can put that into times 10 to the minus 8 or whatever it is to take all the zeros out. Your answer will end up the same, but I've done it like this to keep it all in the same kind of scale. So in order to work out the volume, you do 0.0002 cubed, and that gives you 11 zeros, 8. And then what we need to do is work out, well, how many times bigger is that surface area compared to the volume? And we need to make it to 1 because we always have that ratio to 1. And it's actually 30,000 times bigger. So I mean the surface area is 30,000 times bigger than 1 unit of the volume, which is incredible. The number of zeros is crazy here. But anyway, for why it's much simpler, we've been given the measurement 11. So we know 11 times 11 is 121. That's going to give us the area of one side of the cube. Then we times it by 6. That gives us 726. And our volume is 11 times 11 times 11, which is 1,331. So there we go, 726 to 1,331. Now we need to find out how many times bigger that surface area is than the volume. So we divide 726 by 1,331. And look at that, the surface area is actually less than the volume. So what does that actually mean then? And we need to make sure we've answered the question thoroughly. Well, it means that the surface area is much larger compared to its volume for a bacterial cell. And this will enable diffusion to take place much more quickly as the diffusion distance is much shorter. And of course, you can say it the opposite way round for the multicellular organism and still get the marks. Figure 8 shows a root hair viewed using a microscope. The root hair was viewed at a magnification of times 50. The image length of the root hair X to Y is 43 millimeters. Calculate the real length of the root hair in micrometers. So we need our little IAM triangle, and we already know that I for image is 43 millimeters, but we've been asked to do this in micrometers, so I just like to times it straight away by 1,000. The image is then 43,000 micrometers, and then you divide that by magnification, so you divide by 50. So 43,000 divided by 50 will give us 860 micrometers. A microscope has a ×5 eyepiece lens. Describe how to use this microscope to observe a prepared slide of root hair cells at the magnification of times 50. Well, we need to place the slide on the stage and put the smallest objective lens in position. Move the stage as close to the lens as possible, but don't allow the lens to touch the slide. Switch the microscope on. You could do that at any time, I suppose. And then you can look down the eyepiece lens and turn the focusing knob until the image comes into focus. Then change the objective lens to the ×10, so you're zooming in, and refocus the image. Now, because the eyepiece lens is ×5 and the objective lens is ×10, you are now looking at that at a ×50 magnification. Root hair cells absorb water and mineral ions from the soil. A scientist investigated the rate of nitrate ion uptake by 2 seedlings, and figure 9 shows how the investigation was set up. So we've got our seedlings, we've got a little bit of cotton wool around them, and then the roots are in a nutrient solution containing nitrate ions, and we are bubbling oxygen gas into the nutrient solution on the left and just an unreactive gas, so no oxygen, in the solution on the right. The scientists determined the mass of nitrate ions absorbed by each seedling every 30 minutes for 4 hours, and this table is going to show the result. So if we have a look, we can already see that we have with no oxygen added. Look, they are the same, so that's leveling off there. We can see that the left hand side column with more oxygen added is generally there are more nitrate ions absorbed. Describe the changes in the rate of absorption of nitrate ions for the seedling with no oxygen added. Use the information from table 5. When you see describe, you're basically saying what you see. So we're going to have a look at this table in detail before we look at writing our answer. So with no oxygen added, it's very notable that between 0 and 0.5 hours, the difference is 60 arbitrary units. So that's the fastest rate. And then between 0.5 and 1 hours, we've got a change in 35. Then between 1 and 1.5, it's 10. The difference is 10. And then we've got 10 again. So we've actually got a constant rate here. And then between 2 and 2.5 hours, we've got a change in 5. And 2.5 and 3, it's a change in 5. So that's a constant rate as well. And where we have that levelling off, so from 3.5 hours, there's actually no more nitrate ions being absorbed. And you can see that there is this gradual decrease in the absorption of nitrate ions. The rate of absorption is fastest in the first 0.5 hours and constant between 1 and 2 hours with a difference of 10, and constant between 2 and 3.5 hours. We saw there's a difference of 5 there. There are no further nitrate ions absorbed from 3 hours, so the rate is 0 for 3 and 3.5 hours. And there is a gradual decrease in rate throughout the investigation. Explain what the results in Table 5 show about how nitrate ions are absorbed. Well, because more nitrate ions are absorbed in the presence of oxygen, which is needed for respiration, we can see that energy released from respiration is important in the absorption of nitrate ions. Therefore, even though some nitrate ions can be absorbed by diffusion, many more are absorbed by active transport. Nitrate ions are essential for plants to grow. Describe how nitrate ions are used in a plant to help the plant to grow. Nitrate ions are used with glucose to make amino acids, and these amino acids can then be used to build plant proteins, and therefore that's going to enable the plant to grow. And that is the end of the Trilogy paper. Thanks for watching. I hope you found it useful, and good luck in your exams.